Okay, in this sample problem video, I'm going to try and show you how to map a disulfide bond. And so to make that a little bit easier, so mapping disulfide, so finding the location of disulfide bonds. So to make that a little bit easier to see, I'm going to use A and B to represent random amino acids, C to represent cysteines, and K to represent lysine residues. So in this example, I'm going to show a protein that has intermolecular disulfide bonds that are linking together two separate polypeptide chains. So ABA, CK, ABKA. So that's going to be my first peptide strand. And then I've got a second peptide strand, ACBABKAB. So again, in this example, A and B are any amino acid. C represents cysteines, which are in this case disulfide bonded, so I'm going to show a covalent bond there. And K represents lysine. So if I were to take this protein and cut it with trypsin in the presence of that disulfide bond, I would get peptide bond cleavage here, here, and here. And when I look at that, if I were to run an SDS page gel, it would have 4 fragments. It would have this entire fragment. So I'm running an SDS page in the absence of a reducing agent because we want that disulfide bond to stay there. So I would have this fragment, this fragment, this fragment, and this fragment. So on a gel, that might look like something like this. So this would represent the 11 amino acid fragment, and then 3, 2, and the single amino acid. Now that's, a gel is normally not going to resolve amino acid or proteins that are this small of difference in length, but for this example it will serve. So I would get this type of a pattern. So now if I go back and I take that same peptide sequence, which I have to write out now because I don't know how to copy that picture. So again, A, B, A, C, K, A, B, K, A. The other sequence was A, C, B, A, B, K, A, B. And now if I reduce it, and I get rid of those disulfide bonds, and I again cut it with trypsin, I get peptide bond cleavage in the same place. But now, when I run the gel, these 2 fragments with the cysteine residues are no longer connected. So I'm going to get 1, 2, 3, 4, 5 fragments in this gel. 1 representing 1 amino acid, 2, 3, 5, and 6. So you get a different pattern. And you use these types of patterns to figure out where the disulfide bond is, because you can also sequence each one of the peptides in each one of these bands. So that's how you figure out where the disulfide bonds are located. This process will also work for intramolecular disulfide bonds. So for example, let's take this peptide sequence, A, B, C, A, B, B, C, B, B, A, C, B. And in this case, we've got 3 cysteine residues. And let's say there's a disulfide bond intramolecularly between these 2. You would do your protease digests in the presence and absence of the reducing agent and compare all the different fragments you got out to show that the disulfide bond was between cysteines 2 and 3 as opposed to, say, between 1 and 2 or 1 and 3. So that's called disulfide bond mapping.